Hello friends, hope you all are doing good and staying safe. So I think you clicked this video looking at the thumbnail. So yes, today we are going to discuss about how to score high marks in GCEAL biology structure paper. So today to discuss about this. Firstly, I'll talk about the general structure of the GCEAL biology paper, and then I'll tell you how you can score high marks in structure paper. And finally, we'll take a look at a recent past paper, which is GCAL 2020 Biology New Syllabus past paper, structure paper. I'll be discussing about the first question so that you can understand what I said. All right. So first, we are going to look at the general structure of the paper. So if you take at your advanced level biology paper, it has two parts, part 1 and part 2. For part 1, you will have a 2 hours time limit, and that is not a big issue if you study properly. Then 2 hours is kind of more than enough. In my point of view, 2 hours is more than enough. For part 1, most probably you can even complete the whole paper in an hour. So talking about part 2, it comprises of structure paper and essay paper. Okay. Structure paper, part 1, you have 50 MCQ questions. You all know that. And for structure, you will have 1 hour time limit. 1 hour time limit. And for essay, you have 2 hours. So within an hour, you'll have to answer 4 structure questions. And within these 2 hours, you'll have to answer 4 essay questions out of 6. For structure, you don't have, you can't choose. But for essay, you can choose. You have a choice. So structure paper, so you might think that you have 4 questions in structure and 4 questions in essay. But why do you need more time, nearly double the time of structure for essay? It is because you have to write more than structure. You'll have half an hour for a question. So you'll have to write so many things. So part 2 is, keep in mind that part 2 will be time is constrained because it's only 3 hours, but it's hard for you to finish the paper within 3 hours. So it is very, very important for you all to time yourself. It's not, you have enough time. So just make sure that you time the paper. Even if you know all the facts, everything, you know the book thoroughly, it's important for you all to time the paper. So try to finish the structure paper within the given time limit, which is 1 hour. It is very, very important. For one question, you'll be having 15 minutes. So as I said, for essay, you have so many things to write. But for structure questions are more specific. They expect you to provide specific answers. So make sure that you cut down on unwanted words. Don't incorporate so many words in your structure paper because they'll be asking state 3 examples. So make sure that you just state the 3 examples. And then they might ask you a definition. So make sure that you know the keywords and just apply the keywords and use these linking words to join your keywords, to join your ideas. That's how you have to answer your structure paper to complete it within the given time frame. So I hope you got what I said. With that note, to make you more comprehensible with what I said, I'll take you through a past paper question. As I said, a recent past paper, GCAL 2020 new syllabus structure, biology structure paper. All right. So let's take a look at that. Okay, so let's move on to 2020 AL Biology New Syllabus Structure paper. I thought of choosing the new syllabus paper because hereafter you'll be getting the new syllabus paper. So it's better to get that paper. Okay. So yes. So these are the questions from your first question. You have 4 questions in structure paper, and these questions are from the very first question. Okay. So the first question, A part, the first question is, what is irritability? Okay. So now you can understand that mostly your first question, structure first question is based on unit 2. So it is very important for you all to keep in mind that you should remember the, the initial units as well. So mostly unit 2 questions are the first question. So first question, A part first question is, what is irritability? So here you know that irritability, so you can easily understand and you can easily recall because it is a very simple question, a very simple definition. So irritability is how we respond, our response to a stimuli from both internal and external environment. So the key points are these, which I have made it in bold: stimuli and internal and external environment. Make sure that you remember these key points, these key words or phrases. So stimuli. So where do you get that stimuli from? From internal. You might get it from your internal environment. You know which is internal environment. Internal environment is the environment around your cells. All right? The interstitial compartment, right? So external environment is something apart from you, like surrounding you. Surrounding your body is your external environment. So you might get stimuli from both the internal and external environment. And irritability, your ability. So irritability already has the word ability. So it is your ability to respond. So you get a stimuli from your internal and external environment. And then what do? Like say, for an example, like now it is too cold. You are in an air-conditioned room. It is too cold. So you know what are the physical changes happen in your body? So those are your response. So your ability to respond is irritability. Okay. So you can see the words are limited. You don't write like number of sentences for structure question. So you just have to remember when answering a structure question, you must be on point. You must be more specific. So just what if they ask what is irritability, just write the definition. They are not asking you the examples. They are just asking you the definition. So just write the definition. And this is one key point. Okay. So the next question is about monomers. You all know that pectin and hemicellulose are polymers, especially to be more specific, they are polysaccharides. So polysaccharides are like, to make it simple, are like many monosaccharides join and form polysaccharides. You all know how they form. So pectin, it is galacturonic acid. And then for hemicellulose, it is pentose. You all have a few like examples in your polysaccharide list. So make sure you know all those polysaccharide examples and their monomers. So when you know that, it is a simple question. So it is directly mentioned in your text. Then the next question is NAD plus, NADP plus, FAD. What are the common functions of these coenzymes? So they act as coenzymes, and then they act as an electron carrier. So this is also mentioned in your text. So you all know that NAD plus, NADP plus, FAD, you all know these things. These can be, you all might have, seen these words, seen these enzymes in your respiratory function. Like for respiration, you all need these coenzymes. And you all know that they are electron carrier. So it is very simple to answer this question. So now let's move on to the fourth question. It is about cytoplasmic streaming and chromosome movement. All right. So as soon as you see this, you can relate to a structure, to a organelle in your cells, which is cytoskeleton. That is what they have asked. Which structure performs these above functions? So cytoplasmic streaming, chromosome movement, these are like made by cytoskeleton. Structural components. Now they have asked what are the structural components of the cytoskeleton. So that you all know microtubules. You have three structural components. One is microtubules, tubulin polymers, then microfilaments, actin filaments, then intermediate filaments. These are the three components: microtubules or tubulin polymers, microfilaments or actin filaments, and intermediate filaments. Okay. So now you might think, yes, like cytoskeleton, they have asked cytoplasmic streaming and chromosome movement. Chromosome movement is, you all know, it is during cell division. During cell division, you all know that chromosomes move. For that chromosome movement, microtubules are important. And for cytoplasmic streaming, microfilaments, actin filaments are important. Cytoplasmic streaming can be seen in plant cells. But if they asked you like which structural component is involved in cytoplasmic streaming, then you would say it is actin filaments. And which structural component is involved in chromosome movement, then you might say it is microtubules. But they have asked which are the structural components of this structure of the cytoskeleton. Then you must include all those three, like microtubules, microfilaments, and intermediate filaments. All right. So then the next question is secondary cell wall in plant. Okay. So where is this secondary cell wall in plant is placed? The location. So you all know that it is placed between plasma membrane and primary cell wall. So plasma membrane is the outer layer of the cell. In plants, you have cell wall, but in animals, it is just the plasma membrane. So plasma membrane, and outside that, you might find primary cell wall. I mean, you might find the cell wall. So there you might find between plasma membrane, if I annotate it, between plasma membrane and primary cell wall, you can find this secondary cell wall. Okay, I have just colored it. So between primary cell wall and plasma membrane, you can find the secondary cell wall. So other than cellulose, what are the substances present there? Those are lignin and suberin. You all know they are like insoluble. So yes, That's it. So you can understand how simple the questions are and how direct they are. The questions are very direct. So 15 minutes, you should make use of that 15 minutes and try to answer it within the given time limit. It's better if you can even finish the structure paper within 1 hour, like in less than 1 hour time period. All right. So then you have the next question in B part. The first question is genetic variations in meiosis. Where can we find genetic variations? Examples. So 3 examples. One is crossing over, and then recombination, and then independent assortment. This you all know. Crossing over leads to genetic variation because you all know that there are these chromosomes and they cross over, so that one part of this chromosome might get transferred to the other chromosome. So this is called chromosome. Then you all have this recombination, and then of course independent assortment, that you all know that chromosomes can change their places and placed in the cell, so that when they are divided into opposite poles, it causes genetic variations. So independent assortment is another main reason for genetic variations during meiosis. Then the second question is plant cell pigment that prevents dangerous reactive oxidative molecules formation. This also you can find in your text, and the answer is carotenoids. Carotenoids are the pigments, the plant cell pigments. You have chlorophylls, carotenoids, xanthophylls. So these carotenoids prevent dangerous reactive oxidative molecules formation. You all know that. Then there is this next question. What is action spectrum of photosynthesis? Okay. So what is action spectrum? It is a graph. So a graph should have, you all know it has 2 axes, x and y axis. So you all have x axis and y axis. In x axis, and then you will have this y-axis. So here in action spectrum, what we do is in y-axis we have this photosynthesis, this reaction rate, rate of photosynthesis. So I'll put it like RP. All right. Then you all have this x-axis. There it is the wavelength. You all know that we have different wavelengths coming from the sunlight. So here you have different wavelengths of light. So so here you can see the rate of photosynthesis in each and every point. So like here it could be like, it could be like here, and then it might vary, right? So what are we doing here? In the x-axis we have what do we have? Different wavelengths of sunlight, and in y-axis we have the rate of photosynthesis. So like in each wavelength, how the rate of photosynthesis is differing or varying. So it is a graph showing the effectiveness of different wavelengths of light in stimulating the photosynthesis. Because for specific wavelengths, the rate of photosynthesis could be high. And for another set of wavelength, the rate of photosynthesis could be low. And you all know red and blue are very effective in capturing. Red and blue wavelengths are very good, and they in those red light, blue light, in those wavelengths, photosynthesis reaction is pretty much effective. So That is it. From this graph, it helps you actually. How did you find that which waves are effective in photosynthesis? It is using this graph. While you have different wavelengths, like for each and every wavelength, how is photosynthesis, rate of photosynthesis differing? So by sketching that, you can find like in certain areas, you can see a up and then there's a down. So red and blue waves are effective in photosynthesis. That is how you came to this final conclusion. So action spectrum is a graph showing the effectiveness of different wavelengths of light in stimulating the photosynthesis. You might change the order of the words, but you should get the idea. What is the idea? Effectiveness of different wavelengths of light in stimulating the photosynthesis. You can change the order of words, but you should bring the idea in the paper. Then you all have the fourth question. C4 plants CO2 fixation. All right. What are they asking is, what are the cell types involved in C4 plants CO2 fixation? What are the CO2 acceptors and which are the CO2 fixing enzymes? You all know that you all have mesophyll cells and bundle sheath cells. Mesophyll cells has 3-carbon phosphoenol pyruvate, which can be mentioned as PEP. And then you all have this 5-carbon ribulose biphosphate, which is RUBP. So the enzyme is really easy. PEP carboxylase or phosphoenol pyruvate carboxylase. Then RUBP carboxylase. So you all know PEP, phosphoenol pyruvate, is more effective than RUBP. You all have studied the reasons, and the reasons are very clearly given in your text. So here I want to tell you, like, while you are being very specific for your structure questions, like, you just have to write mesophyll cells, then 3 carbon. It's like, you can mention phosphoenol pyruvate, but it is better if you can mention 3-carbon as well. So like, try to mention 3-carbon phosphoenol pyruvate, 5-carbon RUBP. So see, the answers are very specific. But here you can prepare for a essay question or even for a structure question by thinking like, all right, we have phosphoenol pyruvate and then RUBP. So which is more effective? Why is that more effective? So by preparing these answers, you can prepare for another question. So keep that in mind as well. So then we have this fifth question. How are about two cells tightly connected? So that you all know plasmodesmata, which is especially you can find it in plant cells. You cannot find it in animal cells. You all know it is a type of cell junction. So it is also a simple question. So now we can move to the next part. I think that's it. That's it. Then you'll have this C part. It is mainly about your like about this classification of animals, and then you might get some questions in evolution as well. And there is nothing to explain in that part. You just have to study it and just make small charts, like small charts to keep things in mind. And that's it. That is how you learn that. So yeah, that's it. So far we have discussed how to answer structure questions. You have to be more specific, cut down the words. Then by answering structure questions, just don't answer that structure question. Think of possible alternative ways of asking questions so that you can be prepared. So when you receive your paper, you might get, if you just analyze the past papers, those are just modifications of, past papers. The new papers you receive are the modifications of past papers. So like when you answer a question, think of possible alternative ways and try to answer those questions as well. So you can simply answer structure paper, and also make sure that please wear a watch or something so that you can keep, you can have a look at time. And yes, of course, wear a very basic, simple watch. Do not wear any other automated or even some grand watches. Please don't wear, because they might get your watch. I'm telling this in my personal experience. So wear a very simple watch, a basic watch, so that you can keep in track with time. And then, of course, be more specific. I'm repeating it again and again for you all to keep that in mind. Try to think of all possible ways. So, yeah, the questions are very easy and very direct. So, that's it. That's what I thought of doing. So, yes, thank you so much for listening so far. And of course, good luck for your examinations. And don't forget to subscribe this channel as well, because that if you subscribe only, you'll get more updates on the videos I make. And if you have any questions or any like suggestions, you can feel free to drop that in the comments section. Just mention your feedback in the comments section. If it was good, or are there any ways to improve? And then tell me about any other specific areas you want me to touch. Like if you want me to do any other specific units, you might tell me that as well. I'll try doing that. Then, of course, that's it. And I'm also planning to do, as I said, I just gave you a glimpse that unit 2 is very, very important. So that my next video will be based on that, like the importance of unit 2. So don't forget to subscribe the channel so that you can see my updates. Yes, good luck and bye.